Thank you so much for the presentation. In general, for the last few years, we have been working more and more about macrofauna ecology. And with this, we have more information now about the species distribution within and between Anchialine caves. Particularly, most of my work is in the Yucatan Peninsula, and one of the big questions here is if Cozumel Island is part or not of the Yucatan Peninsula in terms of the Anchialine ecosystems. When we are looking, for example, at the diversity, we see 4 species that are found both in the Yucatan Peninsula and Cozumel Island, including also one marine species that is found in both of these regions. Also, something to notice here is that most of the work that we have been doing has been only with animals, literally just a leaf from the whole tree of life. One of the big questions is what's happening with the rest of Eukarya, but also with bacteria and archaea. So we know that the microbial diversity is very important. They can help us as being indicators for very different things like pollution, but also environmental conditions. They play a very important role in terms of the morphology of the caves. For example, these stalactites are formed by microbial activity. And we know that they have an important link between the micro and the fauna. So, for example, we can set all towards the food chain, and even we have some cases where we have chemiosynthetic bacteria in symbiosis with macrofauna. So the objective of this work is to identify the ecological patterns of the sediment microbial and macrofaunal communities from 2 adjacent Anchialine cave regions, and with this characterize the diversity of the sediment microbial community, determine the continuity into 2 geographically close land masses, and to identify ecological patterns to better understand the diversity and distribution of life. For this particular work, I was comparing 5 caves in the Caribbean coast of the Yucatan Peninsula and Cozumel. The number that you see in parentheses is the number of macrofauna species recorded in each one of the caves. So we have a wide range of conditions, both biologically, but also in terms of the cave morphology. You can see that we have some caves that are very extensive and shallow, others that are short and deep, both with direct connection with the ocean and indirect connection with the ocean. Both of these in both regions, so it is a perfect environment to be doing this type of comparisons. In terms of the microbial community, I was taking sediment samples using the protocol from the Air Microbiome project, so that way it is also comparable with other studies from other environments. In this case, it is using 16S sequencing. And once we have the sequences following this standard protocol, we have one negative control that is everything following the same methods but without a sample. And whatever is in that negative control to the family level that appears with more than 0.5% on abundance of sequences is removed from all the sequences. This to be able to remove some contamination. In terms of the macrofauna community, I am comparing data from 2019, 2021 from visual surveys, in this case of 15 meters squares, in which I am counting and measuring all the macrofauna that I can observe. For safety reasons, areas that are further than 1 hour from the entrance or deeper than 35 meters deep, the transects are 10 meters squares. So with this, we can see that we have a lot of bacteria going on. In general, these graphs are to the class level. You will see here there is a division of the sites and caves from Cozumel and the ones from the Yucatan Peninsula. And in here, something that you can see is that we have a higher number of bacteria than archaea. And when we are looking at these different classes, the first thing that we notice is that several of them are also dominant in sediments from other caves, particularly marine caves in Bahamas and the Eastern Adriatic Sea Islands. But also, these are also dominant in general in marine sediments. So once we start looking into the details of these different caves, different sites, we see, for example, here a higher abundance of these two classes, only in the caves that are small and deep, and something that we see in this case, both of these caves have a hydrogen sulfide layer. When we are looking at what these microbes are doing, they are related with chemosynthetic processes and sulfur cycling. So what we are observing here, it is reflected with the microbial community. When we are looking here, I am presenting La Quebrada. I have 3 different sites. In here you can see the 4 samples, and 2 of them are from site 4. One from 2019, one from 2021. So on 2019, we see what we were expecting, that it was more related to site 3, that is the closest one. But in 2021, it is more related to site 1 that is close to the ocean. So we see this shift in the microbial diversity in this case, and particularly these classes are anaerobic and also related with the phosphorus cycle. Why is so important this observation is because site 4 is the chamber that is the type locality of this ramiped, and it is usually only observed there. You can be diving for hours in this cave, not see a single ramiped, arrive to this small chamber that is at least half of this room, and see 10, 20 ramipeds. Well, in 2021, we didn't observe any. So we don't know if this shift that we see is telling us that these ramipeds are using somehow or interacting with these bacteria, or if both these bacteria and the ramipeds are having the same response to something that happened, that we do not know what. Now, something to mention in here is that when we look at the numbers of classes of Bacteria and Archaea, most of them have a very low abundance. So in this case, 200 classes have only 18% of all the sequences. So one of the big questions here, also when we see bacteria, 22% of them were not classified even to phylum. So one of the big questions is if there is also stegobian microbes, and in such case, if we have a high endemicity rate of the microbial community as we have seen with the macrofauna. But this is something that we need to further investigate. Now, when we are looking into the community structure, I will start first with the macrofauna. And you can see all the symbols that are solid are caves from the Yucatan Peninsula, and all the symbols that are lines are from Cozumel Island. Each different color corresponds to a different cave, and each symbol corresponds to a different site. So I was looking at 30% of the known species from the Yucatan Peninsula and Cozumel, and what we see here is first a clear distinction between both regions. Also, when you are comparing, you can see that each cave is different, and sites within each cave are different too. Something to mention here too is that in the case of Cozumel Island, we have several species that are stegobians, but we also have marine species that have been able to survive inside of the caves, while in the Yucatan Peninsula, most, if not all of the species are stegobians. So when we are asking the same question to the microbial community, we see this little mess. There is not a clear distinction between both regions. So with this, the first conclusion is it depends who you ask. These ecosystems are semi-isolated, but they are not discontinuous. If you ask the macrofauna, they are heterogeneous. If you ask the microbial community, they are more homogeneous. And probably we didn't see this distinction between Cozumel and the Yucatan Peninsula because it is sediment microbial community that probably is moving through the seafloor. Again, it is something that we need to further work on. Something that is very interesting here is the absence of marine fauna in the Yucatan Peninsula. And in this case, what we see is that the territorial expansion and the permeability of the rock could be playing a very important role in here. So in the case of marine fauna, we said in Cozumel, but also in other small areas like Bahamas, Bermudas, and in general islands. So probably something that can happen in here is that this huge mass from the Yucatan Peninsula is accumulating a lot of water that is pushing out. So most of the time when we are close to the coastline in the Yucatan Peninsula, we have fresh water. However, in Cozumel Island, we have brackish water, which is a barrier a little bit easier to jump for the macrofauna, and probably this is why we have marine species in one region but not in the other. In terms of the continental platform continuity, again, we are looking in terms of the community structure, that these two areas are different Anchialine ecosystems, but also in terms of the diversity, and you will see more about this in the next talk. So even when we are looking some of the genus that are highly distributed in the Yucatan Peninsula, they are not present in Cozumel Island, for example. And this has sense even when we are talking about sea level changes, when if we move back 40 million years ago, the lowest it has been is only 100 meters deeper, which you can see is still separated Cozumel from the rest of the Yucatan Peninsula. With this, I just need to thank all the institutions and all the people that has been involved in this project. Thank you.
All right, now how about Fernando? I think we have time for one or two questions, quickly.
So I've done a lot of this kind of work, and what I've noticed more recently is that people are taking the sequence data and they're just bringing it down to the family of the bacteria. And, you know, if you look at animals just at the family level, and let's say you look at rats and different continents, they might still all be in the same family, so you might think they're the same, but they're really completely different. And how do you resolve that with this kind of analysis?
So for sure it's something that we need to be working further on. So one of the main problems is that the region that we are working on is very small. So we can trust until like some level. For sure in the future what we need to start doing is more metagenomics in terms of like having full genome sequences of the microbial community, and that will allow us to go even to a species or subspecies that is tricky with the microbial communities. That also the other problem is in terms of databases. So we will have a bunch of taxonomic units that not necessarily are species, because in a lot of cases we don't have that resolution in general. So there is a lot we can learn in terms of the community structure, but for sure we need to go to a smaller species levels to have a lot of answers. And for example, if there is any Stegobion bacteria and archaea.
Yeah, so if you're at the family level, how do you know if it's a Stegobion or not? Because it might be in the same family or class, but other than that,
yeah, in this case, at this point, we cannot. And that is why I say it's very exciting that now we are having this information, and now we have the molecular tools to allow us to go deeper.
Congratulations for the talk. That was very, very nice.
Thank you.
Given that your question is to see if there is differences between Yucatan Peninsula and Cozumel, have you tried to see if there is shared continuous across the samples instead of just looking at the composition in terms of classes?
So this is one of the works that I come in, like working the most right now. So actually, I just got sequences from the water samples too. So it's something I will be working in this couple of months. But it's going to be very interesting. One of the problems is the amount of information. So when we are looking at species of fauna, we usually work with less than 100. In this case, if we are looking at operational taxonomic units, we are talking about thousands. So it's a challenge, but for me it is something that is really exciting to see the results. Hopefully it's going to be ready before the next conference. Yeah.
All right, thank you, Fernando.
Okay.